right? And this lab afterwards will have a homework that you turn in directly after lab, so pages 19 and 20. But it's also going to have our first lab report, right? Now, when I say lab report, I will explain the components towards the end of this little intro PowerPoint I have here. But it's only going to be 2 parts of a lab report, all right? The introduction and the results, rather than a full introduction, method, results, conclusion, all right? I'll explain that all, but that's what you have to look forward to at the end of the lab here. The lab itself, we're really going to look a little bit deeper into soils and particularly look at these soil simulates. Again, I say soil simulates, even though it should be regolith simulates, right, of Mars and the Moon, all right? So, if you have not had a chance to read through this first part, go ahead and do so. But I am basically going to explain all of this before I let you start the lab here. So I have a little PowerPoint prepared here that is available to you up on Moodle here. And I just want to briefly talk about how we might classify soils, right? I don't expect you to know any of this for an exam, all right? This would be if you took an agronomy class that was solely about soils. Maybe you would want to learn all of this information. But for this class, I feel like it's just some good background knowledge to kind of help you understand how soils might be classified. All right, so if we look at soils, there's multiple classification schemes worldwide, just kind of depending on where you live, right? In the U.S., we use this classification scheme called the soil taxonomy. And when we're looking at classifying soils here, we're looking at unique soil properties. So how are they unique in some way? We're looking at soil horizons, which I'll talk about on the next couple of slides here. And basically, we're just creating a language for us to be able to compare and contrast soils with one another, right? And so when we look at classifying soils, we're going to look at soil texture, right? So we're going to look at the different soil particles that are present and how many sand, silt, and clay particles are present in each soil type. We're going to look at soil profiles and soil horizons. And so when I say a profile, it's basically if you had dug straight down into the soil and pulled up a cylinder of soil, you're going to see some layering going on. Right, that would be the soil profile, and the profile consists of these different horizons or these different layers within the soil, right? And so if we look at, say, this cutout of a chunk of earth here, we can see that it's deviated or classified into all these different soil horizons, right? But we're also going to take into account things like soil moisture and soil temperature, because that can play a role into, how many nutrients are available in these soils. Right, so if we look at these soil profiles and soil horizons, there's very strict language surrounding each of these soil horizons. So for example, the O horizon is always going to be on top. It's going to be a very thin layer here, and that's going to contain all the organic matter that's actively decaying. If we look at the R horizon, right, this is going to be the opposite end of the spectrum here. This is that bedrock, right, that those rocks, those hard substances that have not yet been eroded, right? And then we have everything in between depending on if there's organic matter, if there's accumulation of different minerals. The C horizon is kind of that zone between bedrock and soil becoming soil, right? So we have all these different ways of looking at soil. Now, when we look at soil horizons and soil profiles, right, based on how much of each layer is present, we can then classify our soils into these different hierarchical, I have the hardest time saying that word, classification schemes, right? So basically, at the very basic level, would be a series, right? This is a very broad definition of a soil. You can then define it even further and put it into a family, right? Subdivide it even more specifically into a subgroup, great group, suborder, and finally an order, all right? Oh, excuse me, I said that backwards. Order is the most broad, and then you can divide it out into way more different classifications here. And usually we're really only going to talk about orders because for most people, the series and the suborders and the families are a little less important than the orders themselves. So if we think about the orders, there's 12 of them worldwide. Again, it's based on what do their horizons look like, right? It might be based on the age of the soil, the temperature, the moisture, right? And so we can broadly classify all these soils in the world into these 12 orders. If we look at the U.S. here, right, a lot of these tend to be mollisols or alisols, right? That would be the classification of the soil we see most common here. In the desert Southwest here, we see aridosols, right? It's arid soils. It's you look at the word arid isols, right? It's very arid, it's very dry, a lot of those desert-like soils, right? And again, I really like this image because it kind of shows you a flow chart of how each of these 12 orders are decided, and it's based on the parent material, the vegetation, how old these materials are, the different climates that are present, right? But I don't expect you to memorize all of these. I just want you to know that there exists a scheme to classify soils, right? In lab today, we're not going to classify them into orders, but really we're going to focus on the soil particles, right? What we learned in the lecture part of class today. And we're going to look at how many of these different types of soil particles are present in our 3 soils we'll be looking at today, right? So we'll be looking at soil, right, from the moon, from Mars, all right? These are all simulants, as close as we can possibly get to the real thing, as well as some deciduous soil, right? So they come from deciduous forests, right? Lots of maple trees, lots of trees that will drop their leaves in the winter. Common around this area, right? So the general procedure is going to be putting soil into one of those graduated cylinders, right? You'll put 75 milliliters of soil, and yes, it's weird to measure amount in milliliters because milliliters is like a liquid measurement, but it's easy when we look at these graduated cylinders. You just have to fill it up to the 75 milliliter mark on these graduated cylinders, right? You will then fill that cylinder the rest of the way up to 250 milliliters with water, right? Water you can get from the sink, right? You are then going to cover those graduated cylinders with parafilm, right? So parafilm is kind of like the science version of saran wrap, right? I think it's really fun. You'll have to peel the backing off of the parafilm, which honestly can sometimes be the hardest part, right? The parafilm itself, in order for it to work, you have to give it like a slight stretch, right? And then you'll stretch it over the top of your graduated cylinder, wrap it around the edges like you would like a saran wrap, right? Now this is not going to be a perfect seal, right? Because the next step, you're going to invert the cylinder and mix up the soil with the water, and you'll have to kind of vigorously invert it back and forth. I would recommend you put your hand over the top of this parafilm to reinforce that seal so you don't invert it and then it all ends up on the floor or on the table, right? So you're going to mix all this up, right? Once it's thoroughly mixed, you're going to let it sit for 30 minutes, right? So there is a little bit of a wait time here. 30 minutes, and in that 30 minutes, it's going to separate itself out into different layers based on the particles, right? And on page 17 here, there's a really good diagram of what each particle layer will look like. Because after those 30 minutes are up, right, you will then go in and measure how much of each particle layer are in each of your 3 samples, right? So from Mars, from the moon, and from the deciduous forest, right? At the end of lab then, right, you can end by filling out the assignment, right? Pages 19 and 20. It will have you write a biological hypothesis and a prediction, all right? Typically we do this at the beginning of lab before we even start an experiment, right? But I'm gonna say because we have 30 minutes of downtime and waiting, I would get your experiment going, and then you can start to fill this out while you're waiting, right? It'll ask you to draw in some of the layers that you're seeing in each of your 3 cylinders, and then ask you some questions about it, right? We are actually going to collect the class data in a Google spreadsheet. I do have that linked on Moodle. I will bring it up on my computer too, so if you just want to come up to my computer and enter in the data, that works. We will collect class data. I will generate a graph for you this time around, and you will include that graph in your lab report. So, lab report. Let's talk about that quickly, because beyond just this assignment, right, I'm going to have you do a post-lab assignment, right? Your first lab report, because writing in the sciences is a little different than writing in other disciplines, right? In a lab report, you're typically going to have 4 parts, right? You're going to have an introduction, right? It's going to give you like a broad background into what am I studying, right? You'll have the methods, which details out how exactly you did the procedure, right? You'll have the results, which is where you describe the data that you got from your experiment. And then you'll have a conclusion, which is where you say, my hypothesis was supported or not supported, right? Maybe you're going to give me some likely reasons as to why you saw the trends in your data that you did observe, right? Now for this first lab report, we're only going to do an intro and results, right? Eventually you will do all of these, and for your Martian soil experiment up in the growth chambers, you will write out a full lab report with all 4 components, right? But that one will be written as a group, right? This first one will be written individually here. All right, so let's talk about an introduction, right? In a scientific paper, an introduction is not going to be 1 paragraph, right? It's likely to be like 5 to 10 paragraphs, right? But I want to just condense the amount of writing down, right? And I think that is a really important skill in science, is to write as condensedly as you can, which is very different from other disciplines, right? I want to get to the point, right, as quick as I can, but by including all the information possible, right? So we're basically taking out all the fluff in the writing, all right? So in your introduction, I'm saying about a paragraph. If you decide to do 2, I'm not going to dock you points for that. But I want you to introduce the topic, right? What are you studying, right? What is the significance to studying, the soil particle makeup of these different soils, right? And when you're writing an introduction, you'll likely include sources. So I'm going to ask you to include 1 source, 1 reference, right? I do want you to cite this reference in text, right? So in text would be, parentheses after that sentence. Typically it's like last name and year, right? I'm not going to be picky on how you do an in-text citation, like the formatting, right? Because in science, honestly, there's so many different formats you could use depending on what scientific journal you submit your manuscript to. As long as you do it in a consistent manner, I'm fine with it, right? So cite at least 1 reference. When you're looking at references, be mindful of the references you're looking at, right? Make sure they are academic, they come from a .edu site, or maybe they're from a scientific journal, something that is not like a blog post or an opinion piece, all right? In your introduction, you're also going to state your biological hypothesis and prediction, right? Now you are doing that as part of the assignment you're turning in today, right? I will get that back to you on Monday, and so if I've made any notes on it, you have some time to edit your report before it is due on Tuesday, all right? So that's the introduction. We're also going to do the results, all right? Again, science writing to the point. The results, you're describing what you saw, but you are not saying why you might have seen this happen, right? And so we're really going to describe the patterns and the trends that we are seeing, right? So maybe the Mars soil, right, has a ton of sand in it, right? So I might say that it was X percentage of sand, right? I'm not going to say, so if you don't see a humus layer or an organic matter, I'm not going to say I don't see an organic organic matter layer because of this reason, right? So I'm not going to list the reason behind it, right? In a lab report, the reasoning would become important in the conclusion. In the results, I'm just strictly stating the data, right? I don't care why I saw the data, I'm just listing the data out, right? So you might choose to describe averages, or maybe you might compare and contrast and say the moon soil had twice as much clay as the deciduous soil. So you can use numerical descriptions in that manner, right? I do want to point out that we want to avoid the wording of saying something is significant or correlated, right? So in science, significance and correlation is saying that I ran some sort of statistical test that shows significance and shows correlation. If I did not run a statistical test, which we are not doing in this lab, right, I should avoid those words, right, because those would indicate that I have a statistical test to back that up, right? Within the results, you're also going to include a figure, right, and you're going to reference your figure in the text. So you might say, as we see in figure 1, right, I'm seeing this trend happen, right? Now the figure, I kind of already explained references. I thought my next slide was on the figure here. But again, references, reputable source, cited in a consistent manner. That's basically all this slide is saying here, right? I'm going to skip to the figure though. So for the figure, I'm going to create the figure for you this time around, simply because I want you to focus on the writing, less on creating figures and working with Excel this time around. I'm giving you a kind of a layout of what a figure caption should be, right? So in science writing, when we write a lab report, when we submit it to a scientific journal, we're going to have figures, right? And figures should be standalone, meaning I can look at the figure and the figure caption, so the writing below it, and know exactly what I am looking at without having to read the entire paper, right? Now, I'm not going to know every little specific detail as to what went into making this figure and the data behind this figure, but I can get a sense of what I'm looking at, how the data was obtained for this figure, what our sample size was. So if you remember, we talked about replicates before, and I'll touch on that a little bit in the next slide here too, and what the trend is that I am seeing, right? So I have given you basically a template here, right, that you will copy and paste into your lab report. I highlighted coarse sand and gravel particles because I don't think we're going to graph those specific particle types this time around, and I will update this and put in the new figure in this slide after lab today, right? But it'll look something similar to this. Are there any questions? All right, I know I'm throwing a lot at you. I do have a rubric in your lab manual of what I would expect to see in each section. I will say first time around when I grade things like this, I tend to be a little bit more lenient, make a lot of comments, because you just haven't written a lab report in this manner before. And then the next time around, I'll start to get more picky about points, all right? As I mentioned before, you will have to say what is a replicate, right? And I don't know if you noticed in that figure caption, I had in parentheses n equals and then a blank for what you are going to fill in, right? N basically stands in for your sample size, right? So I have a couple of examples here, all right? One is I analyzed 12 samples, 6 samples per variable, right? My sample size here would actually be 6. I would say n equals 6. Even though I ran 12 samples, in each independent variable, so in each category, there was only 6 samples, right? So essentially, I replicated this experiment 6 times, right? The next example here, I am saying each group analyzed 1 sample for each of 3 variables, and there were 10 groups, right? And so I would say that the sample size is 10 in this case, right? In this lab today, we're going to have 5 groups, and we're each going to run 1 replicate of each soil type, right? So in this case, what is our sample size going to be? I have 5 groups, Yeah, we're essentially repeating this experiment 5 times. Yes. All right. As far as overall composition, right, I'm kind of be a little less picky with this. But I do expect, have some labeled sections. So have a title, have a section labeled introduction, and then your paragraph of an introduction. Have the section labeled results, and then your paragraph or 2 of the results, right? References, long form, cite your references, right? But also make sure to cite them in text. I'm saying typed double space. I'm not giving you a page limit, but rather I'm telling you about how many paragraphs to expect to write. And make sure we're using metric units. So in science, we want to use metric units. So we want to say milliliters instead of fluid ounces, right? Ooh. All right, that was a good ending to my lecture here. All right, that's all I had to say. Are there any questions? All right, this lab report, like I said, is due on Tuesday, so you do have me on Monday if you have run into any issues. But let's go ahead and start doing this. I have 5 stations. I basically put 2 stations on each table, except for this far table here, because I knew fewer people sat there. If you feel crowded or you want to move over to this far table, by all means. Otherwise, divide yourselves into 2 groups on each table, 1 group there. And go ahead and get started. All your materials, soil-wise, is along the back bench, right? When we get to cleanup, I will tell you how I expect you to dispose of the soil. All right.